righty. Did that one. Let's see.
Do we need to know anything on the liquid medium?
Broth is liquid medium.
That's it?
Agar solid medium.
Okay.
On your solid medium, I took a sample from my broth. I streaked it out over the plate. Do I see anything there?
No.
Not much, right? What are CFUs?
Colonial.
Forming
Colony
unit.
Colony forming units. Why are they called colony forming units? They're going to form a colony. But what are they right now as soon as I put them on the plate and spread them apart?
Smear.
They're individual cells. Theoretically, one CFU is one cell. That one cell, put it in an incubator, it's going to divide and divide and divide, 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 divide until it grows into a visible colony. So I can see colonies on my Petri plate after incubation, but when I first use that big surface area to spread out my bacteria from the little drop on my loop into individual CFUs, I'm probably not going to see the CFUs unless I have really good vision, because you need a microscope to see an individual bacterial cell. Even then it's going to be hard. Okie dokie. What do you call it? Say we took my Petri plate out of the incubator, and on my streak marks for the first quadrant, it's just growing all over the streak marks. And in my second quadrant, it's just growing all over the streak marks. But in my third quadrant, I see separate little colonies. What are they called? They're called colonies, but they're isolated colonies.
Isolated.
Isolates or isolated colonies. What if my isolated colonies are all white with a perfectly smooth edge and a shiny texture, and they all look exactly the same? What kind of a culture do I have, probably?
E. coli.
A pure culture, a pure culture. What if when I look at it, I see some yellow colonies, I see some red colonies, I see some white colonies? Now what kind of a culture do I have? A mixed culture, good. What if my professor gives me a Petri plate, and it's just got a whole bunch of everything growing on there? I don't even see individual colonies, so I just stick my loop in wherever, and I mix it into a drop of water, and I make my smear, and I heat fix it, and then I gram stain it. And when I look at it in my microscope, I see some red tiny bacilli and some purple streptococci. What kind of a culture did my teacher give me?
Mixed.
A mixed culture, good. I have two different cellular morphologies there. If I only have one kind of cell, only see tiny red bacilli and they all look pretty much the same, then I have a pure culture, good. Alrighty, don't forget your temperatures. Body temperature is what?
37.
37 degrees C. What if the doctor comes in and he says, oh, you better do something about that patient in the room next door. His body temperature is 32 degrees C.
32?
32?
That's really low.
Put the bear hugger.
It's really low.
It's really very low.
Might be time to toe tag him.
Yeah.
You're not blood play your warm right now.
Probably dead.
He's either got severe hypothermia or he's not metabolizing anymore. Okie doke. What's room temperature?
37.5 or something like that,
No, it's
No.
at 97. No.
Room temperature.
Like
For me,
75
75.
degrees.
I thought.
So
What would it
it'd
be
be
in
like
degrees C?
no? How many degrees
Hold on.
C?
77.
How many in C?
Remember I said if you live in Florida, room temperature is different from down
Yeah.
south. When I set the,
23.
yes.
20
When I set
23.
the incubators for room temperature, I set them to 25 degrees C. That's what we use
25
for
C.
room temperature with our incubators, 25 degrees C. So it's not a cold room and it's not a warm room, it's kind of in between. What is 25 degrees C and F, Colton?
75
I
or
just
something
changed
like
it.
that? No?
37 is 98, you just subtract the difference.
What is it?
37 is 98,
70,
you
yeah.
subtract the difference from that, right? So,
That's a rough estimate.
77.
77.
Yeah, about 77, which is warm. But in Florida, that's what I have my AC set on.
I used to use
77. Right now, it's set on 77. Then when I go to bed, crank it down to 74. Because you sleep better when you're cool.
Mine's at 72.
I did that too.
Yeah.
You go north of the different county, you can go to 64.
Yeah, I know.
I know.
I know. You go up there in the summertime and you're freezing.
I know.
And they don't even have the air conditioner on. I know. Oh man, I hate winter so bad. Oh God. Don't even mind, it makes me shiver just think about it. Okay. Let's see. All right, you will get some questions about catalase negative and catalase positive. All Enterobacteriaceae are catalase positive. Now that's not exactly true. There's some that aren't actually Enterobacteriaceae, but they're in the same section, like Vibrio, and they're catalase negative, but don't worry about that. For the purposes of the test, for the most part, Enterobacteriaceae are catalase positive. However, the test is absolutely unnecessary. It's not even on your API test strip, because a catalase test is not done on Enterobacteriaceae because they're all catalase positive. So I can't tell E. coli from Serratia with a catalase test because they're all going to be catalase positive. It's not helpful when you're trying to differentiate Enterobacteriaceae. I'll tell you what a catalase test is used for. Cocci, Streptococcus, and Enterococcus are catalase negative. Staphylococcus is catalase positive. Strepto, entero, negative. Staph, positive. Catalase positive. So what does that tell you about Enterococcus and Streptococcus as compared to Staphylococcus? They're more anaerobic.
Staphylococcus and what's the other one?
Staph is catalase positive.
What was the other one?
Staph is catalase positive. Strep and Enterococcus are catalase negative. By the way, Micrococcus is Staphylococcus. It's the same thing. It will be catalase positive. Some books don't even use the genus name Micrococcus. They call them all Staphylococcus. Remember Micrococcus forms clusters of cocci, often in tetrads? Those are Staphylococcus. So some might call them Staphylococcus luteus instead of Micrococcus luteus. So remember, Micrococcus is a Staphylococcus. Enterococcus is Strep D, remember? Enterococcus is actually a Streptococcus. I know, it's also confusing. Micrococcus is a Staphylococcus.
And Enterococcus
And
is a
Enterococcus is a Streptococcus.
So Streptococcus will be negative, catalase negative, right?
Right.
Don't
Alrighty,
laugh at me.
let's go back to the catalase test again. I put hydrogen peroxide on bacteria. If they're catalase positive, they bubble, they break down the hydrogen peroxide. Why do they want to break it down? Because they don't want to be harmed by it. If they can't break down the hydrogen peroxide, they will be harmed by it.